Okay, so let's just get going. We are back to the male reproductive system right where we left off, talking about scrotum. So I left you to sort of ponder this question about why human testes are located external to the body, why we would store such an important reproductive organ in such a vulnerable location, and sort of the one reason that we can figure out or that we know all has to do with temperature. So the testes themselves are really adapted to live in a cooler environment than what we find in the inside of the body. So our core body temperature is around 37 degrees Celsius, right? And the problem is that we cannot, as humans, produce sperm anywhere above 35 degrees Celsius. And so that might not seem like a very big difference, 37 to 35 degrees Celsius, but that is enough that if a testis is internal, it will not produce viable sperm. So the sperm are going to be very picky. They are going to be very specific, and they are not going to survive at anything above 35 degrees Celsius. So we cannot, therefore, keep the testes internal to the body because they would be this temperature, or maybe even a little bit higher, but they would not be able to produce sperm for reproductive purposes. So we are going to keep them outside of the body in a sac that allows them to be a little bit cooler. We do have 3 mechanisms that are used to regulate the temperature of the testes, and so I'm going to go on to the next slide because those are outlined there. So our 3 temperature regulation mechanisms are going to be, one is the cremaster muscle. So the cremaster muscle are essentially strips of the internal abdominal oblique muscle. So if you don't remember that muscle, take a look back at it, but we have some strips of that here that are going to sort of wrap around the spermatic cord, and when they contract, the testes are going to move up and down. So we can see in this, we have sort of these long muscles here that are wrapped around the spermatic cord, and so when they contract, they're going to contract this way, they're going to get shorter, and they're going to bring those testes up closer to the body. So when the testes move upward, they are going to move towards that warmer temperature, and they will therefore warm up. So if it's too cold in the testis, if it's too cold, sort of the skin of the scrotum is loose and they're hanging fairly far away from the cavity of the body. If you were in cold water, if you're outside in the cold temperatures, these muscles would contract and pull those testes up and nestle them much closer to the body, where they would therefore be warmer. Next, we have the dartos muscles. So the dartos muscles are a subcutaneous layer. And so when I say subcutaneous, if we look here, we can see our tissue, and then we have this thin layer of muscle underneath here. Those are going to be the dartos muscles. So the dartos muscles are going to contract again in the cold. And when they do that, they are going to change the surface area that's present on the outside of the scrotum, and it is going to reduce the amount of heat that's lost to the surface area. So this is changing whether or not the scrotal tissue is sort of flat and stretched out, or whether it's tauter and has sort of a more wrinkled appearance to it. And then the third mechanism is going to be what's called the pampiniform plexus. So the pampiniform plexus is a network of veins. And so we can see in here we have basically one testicular artery, and then sort of wrapped around a lot of the structures in that spermatic cord, we have this network of vein. So it's a venous network. It's countercurrent. So the blood that is going to be exiting in the pampiniform plexus is about 1.5 to 2.5 degrees cooler than the blood that is at body temperature. So let's see if we have Okay, so we have blood that's cooler than core temp leaving or exiting the testes. The blood that's coming in via our testicular artery is going to be core temp. Okay, so we have very warm blood coming in the testicular artery. We have much cooler blood leaving out the pampiniform plexus. So this is our countercurrent system. Maybe if you don't remember exactly what that means, you should go back and look at our urinary system where we talked about our countercurrent exchange. But so out of the body, we have blood moving in one direction or out of the testes. And then into the testes, we have blood moving in the opposite direction. So these are countercurrent. Counter means opposite and current means flow. So we have opposite flow of the blood. And what happens is as this warm blood comes into the testes, some of that heat is going to actually get picked up by the pampiniform plexus. So it actually absorbs the heat from the testicular artery so that by the time the blood is approaching the testes, it is much cooler. So it actually gives its heat to the pampiniform plexus and it cools down the blood that's entering the testes in the testicular artery. So what we don't want to happen is we don't want the testicular artery to be bringing 37 degrees Celsius blood directly to the testes, because that temperature is going to warm up the testes and it is going to cause damage or issues with sperm production. So we need to cool this blood down before it actually reaches the testes. So what we do there is this countercurrent exchange. And so this blood, I wish I was better at drawing, but it would cool down. Like as it approaches the testes, it would go from really warm to cooler to cooler to cooler to cool in terms of body temperature. All right, so these are our three mechanisms. Our cremaster muscles, so pulling up and down or into or out of that pelvic cavity almost. Dartos muscles, which is going to change the scrotal tissue and how much surface area it has. And then the pampiniform plexus, which is going to give us countercurrent heat exchange with the incoming warm testicular artery blood. Now looking at the testes, which are internal to the scrotum, these are going to be endocrine and exocrine glands. So endocrine tells us that they are producing and secreting hormones. And then endocrine tells us that there is a product being secreted via a duct, which in this case is going to be sperm. So we have both endocrine and exocrine functions. Okay, and we will talk about both of these different functions in this lecture. So they're about 4 by 3 centimeters oval. Okay, and so they're sort of slightly flattened. They are covered in a couple layers of connective tissue. Remember, we have this tunica vaginalis. The tunica vaginalis is going to come from that invagination of the peritoneum, or not peritoneum. Yep, maybe that was right. Peritoneal membrane. There we go. Coming from the abdominal cavity. And then we also have a layer of connective tissue called the tunica albuginea. The tunica albuginea is actually what gives the testis that really almost pearly white color on its outside surface. So we will see that sort of over and over again. Inside of the testis, we are going to have septa. So we get connective tissue that sort of moves in from the outside and connects, or divides rather, the inside of the testis into lobules, or into these sort of wedge-shaped units. Those wedge-shaped units are going to contain 1 to 3 seminiferous tubules each. So each, each of these lobules is going to have 1 to 3 Now, we will talk about seminiferous tubules here in a few minutes, but seminiferous tubules are going to be the actual site of sperm production. And so each of these is about 70 centimeters long. So these are really long, sort of twisty, convoluted tubes. You can see them over here, all sort of wound up. But so each of these lobules, and you can see here 1, 2, so this lobule would contain 2 seminiferous tubules. Looks like maybe this one, 1, 2, 3, would have 3. But we have 1 to 3 of these really twisted, sort of long, very skinny tubes, which is where we are going to actually do sperm production. Okay, and so I think that might be all I say here. Let's just sort of look. Here's our spermatic cord, right? And it's going to be sort of covered. This is that cremaster muscle here. Inside, we have our tunica vaginalis, and then right up against the testis, we're going to have the tunica albugenia. Inside, we have our seminiferous tubules in those lobules, and so these sort of division pieces here are the connective tissue moving inwards and forming lobules on the inside. We will talk about this again later. We'll see this pathway, but we have our seminiferous tubules, and these are really narrow, long tubes that produce sperm. Those sperm are going to get pushed out of the seminiferous tubules and into what's called the rete testis, which is along this posterior side of the testis, out through these efferent ductals into the epididymis, which sort of curls around. We have a head, a body, and a tail. It then flips up and becomes the ductus deferens up through that inguinal canal. So I already said a few of these things, at least the first couple of bullet points on here. This is looking more specifically at the histology of the testis, so looking inside at what's going on in these tissues. So in the testis, we do have these seminiferous tubules. Main point here is that the seminiferous tubules are producing our sperm. They have a lumen. So remember, a lumen is an open space within a tube. So if you're looking down a garden hose, that hole in the middle would be the lumen. Same thing here. So if you imagine this is a garden hose, this is going to be the lumen or the open space. We have lining the edge here, so we can see this sort of circular edge. These are going to be germ cells. So the germ cells are going to be the cells that will give rise or form the sperm. We also have sustentacular cells. Sustentacular cells are sometimes still referred to as Sertoli cells. I will only use the term sustentacular. But sustentacular cells are sort of located in between some of these germ cells, and their job is really going to be for nourishment, waste removal, and provision of growth factors to our germ cells. So they protect and nourish the germ cells. They're like the nannies of the germ cells in this seminiferous tubule. Located in between the tubules, so we have one seminiferous tubule here, and you can see we have the outline of another one here and the outline of another one here. In between the different tubules, we have what are called interstitial cells, and this time I'm going to call them interstitial cells and not Leydig cells. Interstitial cells are going to be the testosterone producing cells. So they are really going to produce the testosterone, which is one of the major jobs. This is going to be the endocrine function. This is going to be the exocrine function. So within the testis, we get both sperm production inside of the seminiferous tubules, which is our exocrine function, and then testosterone production in those interstitial cells, which is between the seminiferous tubules. That's going to be our endocrine function. We do have a very interesting blood testis barrier that's located in the scrotum, or in the testes rather. This barrier is going to prevent immune cells from attacking the germ cells. Now, the germ cells, what I'm talking about here, are going to essentially be the sperm and some of these last stage spermatids or cells that are about to become sperm. They are genetically different from the parent cell. So here we have a cell that is going to be 46 chromosomes, the exact same complement as what we have located in this Leydig cell, right? Or a skin cell or a neuron, whatever it may be. And so the immune system recognizes these as self cells and is happy with leaving them there. The problem is these sperm are different. They only have 23 chromosomes. The combination of chromosomes is different. They no longer look like the parent cell. So we must separate the sperm from the immune system, because otherwise immune cells are going to come in and destroy all of these sperm, and we're not going to have any reproductive cells. So we have a nice tight junction that gets formed by our sustentacular cells. Okay, so we get tight junctions here, which is going to keep the immune system essentially on one side and the sperm on the other side. I do believe that there is some evidence that if the blood testis barrier is weak, or if the immune cells are capable of penetrating that, that could potentially be a cause of low sperm count or infertility, as the immune system essentially eliminates all of those cells. So we need to protect the sperm because they are genetically distinct from the rest of the body, so they don't have the same protection from the immune system as the rest of our cells do. Okay, I am going to end this little mini lecture here and come back to talk about our duct system in a few minutes.